Hello, I'm David Field and I'm making this YouTube recorded version of my PY1BP lecture so that people who need to self-isolate or otherwise stay away from the lecture theatre because of the coronavirus can watch the lecture. At various points in the slides I actually refer to other YouTube videos, and I can't embed those in a playing form or clickable form here. So in order to watch those videos, what you'll need to do is download the PowerPoint slides themselves from Blackboard, and either on the slides themselves or in the notes underneath the slides you will see the link to the relevant video. And note that I also put quite a bit of more information in the notes section that you can read. Today's learning objectives are mainly, the biggest one is to cover the basics of colour vision, but also we're going to take a look at the Milner and Goodale two visual systems theory and get you to know a little bit about myopia, which is short-sightedness, and amblyopia, which you may know of as when a child has one lazy eye that requires them to wear a patch at school. All of the learning objectives for today are further supported by reading chapter 10 of your Breedlove and Watson textbook. So this is the first slide about colour perception or colour vision. And the first thing to say is that before we can understand anything about how we experience or perceive different colours, we've got to take a little look at the physics of light and of coloured light. In fact, to understand colour perception, you're understanding the interaction between physiology and physics. So the first two slides will essentially be physics. What this graph is showing is a breakdown of the spectral composition of white light. Now, white light is the sort of light that you would have during the midday sun. It's the sort of light that probably in a physics class back in high school you shone white light into a prism, and the prism split the white light, producing a rainbow pattern on the other side of the prism, which is always very beautiful to see. Now, of course, light is made of photons, and there's no such thing as white photons or white light itself. White light is what you get when you get an equal mixture of all the other wavelengths of light. And the wavelengths vary from what we call short wavelength light to long wavelength light. And at the top of the image there you see violet light, which has the shortest wavelength in the diagram, 400 nanometers. And what that means visually there is that the peaks and troughs of that waveform are closer together. And as we move further down, you can see that the peaks are further apart, so that's a longer wavelength. Those numbers indicate the actual wavelengths of the different colours of light, from 400 nanometers for violet at the top to 665 nanometers for long wavelength red light at the bottom. Now, the other bit of physics we need to consider is something to do with the properties of surfaces that light can reflect from. Every surface in the world has a property called its reflectance, which allows you to plot spectral reflectance curves, where basically the y-axis of the graph tells you the percentage of light that the surface reflects versus absorbs, and the x-axis is the wavelength of light, that is the property that I showed you on the previous slide, going from the short wavelength violet light up to the long wavelength red light. So we look at some of the lines on this graph. The one labelled white shows that a white surface reflects equal amounts of all the different wavelengths, and it pretty much reflects all the light. Down at the bottom, the black surface reflects only a very low percentage of each of the wavelengths of light, absorbing the rest. But again, it's an equal amount of each wavelength that is reflected, and because it's absorbing so much light, that's one reason why black surfaces tend to heat up in the sun. Now clearly, it's fairly intuitive that the red surface reflects more of the red wavelength of light and absorbs the other wavelengths, and a similar picture for the green and the blue surfaces depicted in the graph. The grey surface is again reflecting approximately equal amounts of light at each wavelength, but an intermediate amount in between the white surface and the black surface. The white fluorescent surface is a special, unique one, which actually reflects or almost emits more short wavelength blue light than is shone onto it, and that's because it's able to convert a little bit of the other wavelengths of light into that short wavelength, and hence it appears to sort of glow slightly when you see it. So that's an unusual one. Now, so far it seems a fairly simple picture, doesn't it? But let's go back to something I said earlier, which is that, you know, at midday, light from the sun is white light. It is made up of equal amounts of each different wavelength, but at other times of day it's not. So for example, in the evening, light from the sun has a much higher percentage of the long reddish wavelengths than it does of the other wavelengths, but the properties of the surfaces the light reflects from have not changed. So that would suggest, wouldn't it, that the grey surface, rather than having a flat line in terms of the percentage of the different wavelengths that are reflected from it, would now be reflecting back a higher percentage of red light because, you know, there was more red light shone onto it relative to the others. And yet we don't perceive grey surfaces as different coloured in the evening to how we see them at midday. We still perceive them as grey. And this is an achievement of human perception of the human brain called colour constancy, which later on in the lecture I'll explain how this amazing thing is achieved. Now, we're now moving on to the physiology and also the psychology of colour vision. So how is it that humans can perceive a psychological correlate of coloured light? Because not all animals can, and indeed not all humans see as many colours as others, as you'll know from the example of people who are colourblind. So how does it work for the majority of us who have the normal system of colour vision? So the graph I'm showing here is similar on the x-axis to the one on the previous page, in that it's going from the short wavelength light on the left to the long wavelength light, the reddish ones on the right. But now the vertical axis, the y-axis, has changed. Rather than reflectance properties of a surface, it's now the absorbance property of photoreceptors. So the absorbance property of a photoreceptor is for a particular frequency of light, how efficient is the photoreceptor at absorbing that frequency. So if we take the example, now you probably know we've got three different types of cone which are responsible for our colour vision, the S cones, M cones and L cones. And you probably know that we also have rods which are responsible for our ability to see in low light levels but don't contribute to colour vision. So if we take a look at the absorbance of the rod system, Well, that is the dashed line on the graph, and you can see that it has a peak ability to absorb light that has a wavelength of 495 nanometers. It's quite broadly tuned though, so it can actually absorb light from below 400 nanometers up to 600 nanometers. And the reason that rods don't contribute to colour vision is simply because the way the visual system is wired up, they're not connected to the wiring of the cones. In principle, rods could work to contribute to colour vision, but they don't. Instead, because they're sensitive to lower levels of light, they're used to give us our night vision. So, looking at the profiles of the three types of photoreceptors on there that do give us colour vision, the cones, you can see that the S cones on the left have a peak that's quite far from the other two types of cones. They peak at 420 nanometers and absorb light up to just under 500 nanometers. Now the M cones and the L cones are actually fairly similar, aren't they? With the M cones having a peak at 530 nanometers and the L cones at just 560 nanometers, not far away. And you can see this sort of triangular area formed by those two overlapping distributions where they're similarly sensitive, but nonetheless there is some separation between the distributions of the M cones and the L cones in terms of what they can absorb. But one thing you should take away from this graph is that the tuning curves of the individual types of photoreceptor are very broad and non-specific, so they don't give you very precise information by themselves about what type of light stimulated them and caused firing. Now, what I'd actually like you to do at this point, if you're seeing this online rather than in the lecture theatre, is watch that YouTube video that I've got the link to there. So you'll need to probably download the one from the Blackboard and click on it, because that YouTube video will kind of give an overview of both the physics and this early bit of the physiology to help you understand the overall story. So when you've watched that, come back, and one thing I'd like to draw out from that video is a comparison with one of the previous lectures about auditory perception, because we saw that the physical stimulus for auditory perception, which is pressure waves travelling through the air, the sound, if you have two different pressure waves emitted at the same time in the same room, they're going to mix up with each other. The waveforms effectively add together into a single waveform. But what you will have seen in that video there is that that's not the case for light. If you have some photons present that have the wavelength that is the long wavelength red light and some other photons present with the medium wavelength green light, they don't mix up and become a different kind of photon. They remain separate, both present at the same time, which is an important distinction. So one question that people will often have is how we can perceive more than three colours. If we have three types of cone, the short or blue, the medium or green, and a long or yellowy red cone, and based on a kind of principle of labelled lines, which we've seen is the principle that applies in some of the sensory systems that we have, you'd only be able to perceive three colours. But the reason we can perceive a lot more than three, in fact almost an infinite number of colours, is because a particular wavelength of light falling on the retina will tend to drive two or even three cones each to a different degree, and that unique combination is a signature for that particular perceived colour. So in this graph, the example is that we shine some light with a wavelength of 580 nanometers that corresponds in physical terms to yellow. We shine that on the retina. The retina has no specific cone that peaks its absorbance at that frequency. However, what you get is a moderate amount of activity in the M cones and a quite high amount of activity in the L cones and none at all in the S cones, but that combination is unique to yellow. Except it's not quite unique, because you actually can produce the same response from the system with a different type of light. So what we've seen so far is that individual colours are defined by particular combinations of response across the three types of cone. But that implies that the brain is got some circuitry to compare the level of response in, for example, the M cone with the current level of response in one of the other cone types. And that circuitry has been quite extensively investigated. And so we're going to take a look at it in the next section of the lecture. Now, essentially, the circuitry that underlies this is two channels, two colour channels, each of which is based on a subtractive opposition of two primary colours. So we'll see what that means first by looking at the evidence that early psychologists in the 19th century used to prove that there was an oppositional two-channel system underlying colour perception. in addition, there is a brightness channel that allows you to mix in the amount of white, if you like, into the colour you're perceiving. And also on this slide, I will say that as well as this opponent system allowing us to perceive the primary colours, which are the ones you see when you split white light with a prism, there are of course other colours, secondary colours like brown, which don't occur in the rainbow and don't occur when you split bright light with a prism, and there is therefore no actual wavelength of light that corresponds to secondary colours such as brown at all. They don't physically exist. They are purely psychological constructs, and yet they are still defined by particular combinations of response across the three basic colour sensitive cones. So they really are an invention of the mind. Okay, so one of the first bits of evidence for the existence of an opponent system was produced using the methodology of adaptation that we looked at in one of the previous lectures. So what I want you to do on this slide in a minute is to do an adaptation experiment on yourself, and we're focusing particularly here on those turquoise greenish bars. We know that if we stabilise that image on the retina, any neural mechanisms that are specifically tuned to those turquoise greenish bars are going to get fatigued after a while. That's the process of adaptation. So let's do that now. Let's stare fixedly at the red square in the middle of that flag for about 15 or 20 seconds, and then I'm going to switch to a blank white slide, and when I do, I just want you to look at the middle of the blank white slide and keep your eyes still for a few seconds. It will take a few seconds for the effects to become apparent once we switch to the white slide. So now I'm already keeping my eyes still. I'll do that as well, and I'll just stop talking. The next two slides are going to present different type of evidence that again was popular in the 19th century and quite decisive in setting the pattern of colour perception theory. So this is based on observations that if you mix two different wavelengths of light, you get different things depending what wavelengths of light you're mixing. So the top bar in this slide on the left hand side is just got 100% short wavelength blue light. On the right hand side it's 100% long wavelength red light, and then as you move closer to the centre, those two sorts of light are being mixed by different percentages. So it's just two wavelengths but with different mixes. But we've already seen that the brain will interpret those mixes of a pair of wavelengths as if they were pure wavelength light, types of light of different wavelengths. Yeah. So anyway, what you see as you move towards the centre of that blue-red bar is that what you perceive in the middle is purple. Now, purple is kind of bluish-red, really. So that mixes and produces purple. Now, the bar at the bottom has got, again, the short blue wavelengths at one end, but now it's got another wavelength at the other, which is yellow, the wavelength for yellow. And these two don't behave in the same way when you vary the proportion of them. In the middle, we don't get a third colour. It just doesn't happen. It just kind of transitions from blue to yellow. So that was evidence that blue and yellow must be opposed within this oppositional system. Now, in the examples on this slide, we've got yellow wavelength light about 580 nanometers at the left hand side of the top bar. We've got long wavelength red light on the right, and when they meet in the middle, what you get with a 50% mix of yellow light and red light, you get kind of orange. So they mix in a smooth way. But in the one on the bottom, the green wavelength light on the left and the red wavelength light on the right, when they mix by 50-50, you get the percept of yellow. So that indicated that green and red are opposed primary colours. Now, it could be that this doesn't work properly on your computer screen because computer screens won't be reproducing the wavelengths exactly in the way that is done when you do this as an experiment in a lab. Now, in the colour opponent processing system, the first stage of opponency happens at the level of the retinal ganglion cells. There is a second stage of opponency that we'll talk about later in the brain, but the first opponent process is instantiated in retinal ganglion cells. Now, the opponency is of course based on the primary colours, and one channel opposes yellow and blue, which are two of the primary colours, and the other channel opposes green and red, which are the other two primary colours. The third channel here, shown at the bottom of the diagram on the slide, is a black-white or luminance channel, which allows the brain to know kind of brightness value of colours. And you can see on the left of this diagram the three cone types represented as those triangles, and you can see that the brightness channel actually gets its input just from the red and green cones, i.e. the long wavelength and medium wavelength cones. It doesn't get any input from the short wavelength cones, and that's actually the reason why our visual acuity is not so good for scenes that are illuminated in a very bluish light. Also, we have less of the blue cones, which also contributes to that effect. But that was an aside. So if we look at the kind of wiring diagram for producing the yellow-blue opponent channel at the top right of the diagram, to produce the yellow, we know we don't have a yellow cone, but we've seen from earlier slides that we can produce yellow by looking at the difference between the red and the green. So that's why both the red and the green cones are in that wiring diagram pointing to the yellow-blue opponent channel. They're used to produce the yellow, and then that's opposed to the blue. And some of the ganglion cells, of course, retinal ganglion cells have the centre surround organisation that we discussed in the previous lecture. So you're going to have some retinal ganglion cells where the central region of the receptive field is responding to blue and the outer region yellow, and then you have some that are the opposite. And then in the other opponent channel, the blue-green channel, red or short wavelength is opposed to green, but with a little bit of the blue added in too. Okay, this graph shows the results of an experiment in monkeys where electrodes were used to record the spiking rates of cells in the monkey's lateral geniculate nucleus. Now that's actually one stage further in the visual processing hierarchy than the retinal ganglion cells, but the properties of the spectrally opponent cells in the LGN are very similar to those that are found in the retinal ganglion cells. So in the top left graph, what we're seeing there is the response of a cell when different wavelengths of light are shone into its visual receptive field. Much like we saw for the earlier graphs where I showed you the absorption spectra of the three different cone types and the rods. So it's a very similar sort of graph really, particularly the x-axis is identical, so you can actually rewind the video and compare. And what this is, it's an opponent plot, it's a cell with opponency between the M cone and the L cone. So it's like subtracting the activity level of the M cone from the activity level of the L cone. Sorry, that's correct, yeah. So because the L cone is the one furthest to the right, the one most sensitive to red, and the M cone is actually quite close to it, quite similar to it, it actually subtracts off most of the response caused by the light to the L cone, and you get this quite nice narrow response such that this cell in the LGN only really makes any response at all to light between 600 and 700 nanometers in wavelength, i.e. the kind of red to deep red portion of the spectrum. And that's a much more narrow tuning than was the case for any of the three cones. So this is therefore far more useful to the brain in a sense, because if this cell is firing, it tells you that there's some red, right? There's some light of that wavelength falling into that receptive field, which is, quite a narrow and useful bit of information. On the top right hand side of the graph is kind of the opposite calculation. It's a cell that has the response of the long wavelength cone subtracted from that of the medium wavelength cone, and so this one has no response at all now in the 600 to 700 nanometers range on the x-axis, but will respond to anything in the kind of light blue greenish part of the spectrum. The two cells shown on the bottom left and bottom right here are a little bit more complicated examples of opponency. The one on the left is getting increasing its firing rate when there's either the long or medium cone is activated, and this kind of what's being subtracted is the blue cone, so it suppresses the response in that region, producing a kind of strong peak in the red region, but not as far into the red region as the one in the top left. And so if we then do further subtractive opponency processing later in the brain between the top left and bottom left cell, then we could imagine how you might get a cell in the brain itself that was kind of very tightly tuned just to respond to very specific shades of red. So that's kind of the principles of opponency. So this slide just starts to get into opponent processing in the brain, which occurs mainly in visual area V4. And what it does is it actually compares what colour perception might be like, what we might be sensitive to in terms of colours, if we only had our cones on the top row, then what type of thing we get through opponency in the retinal ganglion cell or LGN level on the second row. You can see it starts to get more of the kind of yellowy oranges emphasised there and a bit more subtlety in the green region. And then in V4, where there are further stages that further oppose the opponent cells that we already built in the LGN and the retina, you then get further opponency, which kind of starts to build in things like that purple that's appearing on the left. So it's a case of refining things in the brain using the same wiring and computational mechanisms that were used earlier in the retina. While one of the things the brain does to contribute to colour perception is to complete the opponent processing that began in the retinal ganglion cells, another thing that it does is a thing called colour constancy. So ask yourself, why do the colours in image A on the left appear the same as image B on the right? Well, if you remember what I said at the start of the lecture, this is quite remarkable because image A on the left is shown illuminated by light of the yellow wavelength, whereas image B on the right is shown illuminated by light dominated by short wavelength blue light. This is like the example I gave of sunlight containing a lot more long red wavelengths in the evening than it does at midday when it contains an equal mixture of all the wavelengths. And because if you illuminate a surface with light that's got more long blue wavelengths, it's going to reflect more blue. It's sort of hard to see why all the squares on those cubes in the images don't appear more bluish when illuminated by blue light, and why you can still indeed see and name colours like red and green and yellow at all. Now, this is the process of colour constancy, and the part of the brain that's most involved in that is a visual area shown in the diagram on the bottom there called visual area V4, which is specialised for colour perception. And what is happening in V4 is that part of the cells there are dedicated to processing and detecting the actual wavelength of the illuminant. So the brain is processing the nature of the light source that's illuminating the scene, as well as the objects in the scene. And we're not consciously aware that it's doing that very often, but in order to adjust our colour perception, the brain has to estimate the illuminant. And then once it's done that, it can subtract the illuminant from the input it's getting from the opponent processes in the retinal ganglion cells in order to make this adjustment and allow us to see the colours of surfaces regardless of the type of light that's illuminating the surfaces. And that's what we mean by constancy in vision. We mean being able to perceive a property of the world independently of variation in what the information on the retina is. So if you would download the copy of the PowerPoint slides on Blackboard, you'll see that there's a link in the notes section to a YouTube video about colour constancy that would be worth watching, because it will further summarise and to give you more information about this. So one of the things you should know about all the constancy operations in perception is that none of them work perfectly. The same is true for things like size constancy. There will be some circumstances under which the brain's ability to allow you to perceive the actual physical reflectance property that is the object's colour, regardless of the type of light that's illuminating that object, will break down and produce mistakes. And we're going to look at one of those now. So one of the features of the brain's colour constancy mechanism is that it operates quite locally on the retinal image. It's not a calculation of the kind of overall average illumination in the scene. It operates very locally, and that's useful generally because the actual type of illumination that's falling on an object, it can vary quite a lot within a single scene, for example, due to shadows. And so here is an example involving shadows that actually fools your colour constancy system. If you look at the cube on the left, you'll see there's on the top panel there's a brown square, and in the bottom panel that faces sort of towards you, that's got the shadow on it, there is an orange square in the middle of that panel. But actually the reflectance of the brown square in the top of the cube and the orange square on the front of the cube is the same. They are physically the same, and they appear different because of the shadow and the fact that your constancy mechanism is localized. And if you look at the image on the right, you can see that a kind of white circle has been drawn in order to block out the shadow and the other background, and now it appears that both squares are brown. And that's because we're no longer getting the effect of the shadow confusing the system. So it kind of removed the illusion, if you like. And here on the next slide, I'm going to have a little bit more of a dramatic demonstration of that. So here we can see that if I make this white polo mint shape appear around the orange square, that it actually causes the orange square to be perceived as brown, just like the one above. Now, if you don't believe me, if you think I'm just cheating by drawing a white square with a little brown square inside, a brown circle inside it, then what you could do is print out this image, and you'll still see the illusion in the printed image, not with the polar mint shape, just print it out, and then get yourself a piece of paper and make small holes in the piece of paper in two positions, one corresponding to the top brown square and one corresponding to the bottom brown square, and sorry, bottom orange square it would appear to be. Put the holes over, and you'll see that when you look through those little holes that obscure the shadow and all the other background, both squares will indeed appear to be brown. Now, another interesting finding in colour perception is that it's not a purely perceptual process. The way that our brains build our perception of the world is not just based on the input from the retinal images plus some low-level mechanisms to adjust for things like the type of ambient light that's illuminating the scene. There are also quite high-level cognitive processes that can influence how we see the world, and this applies to colour as much as any other aspect of perception. And our memory of what objects typically look like can influence in a sort of subtle way how we see a black and white image. This has been shown recently in some high-profile publications. Now, this image is one that's made up of variation in the black-white channel, so it's a grayscale image, and the only other variation is variation in the amount of long wavelength red light. There is no blue or green light emitted from this image and no yellow light. And yet you can see that the image on the right is clearly a yellowish looking lemon, even though there's no actual normal visual stimulus there to produce that percept of lemon yellow, i.e. there's no wavelength of light that actually corresponds to yellow, and neither is there the mixture of red and green light that the brain also typically interprets as yellow. The reason you're seeing that as slightly yellow is due to your prior memory information about the different fruit. So, however, just to compare what that kind of unusual percept looks like with the normal one, where all the different wavelengths you expect from the apples and grapes and lemons are now actually being emitted from the screen at you, we can see, of course, that it's a far more vibrant image when the colour perception is based on more than our prior kind of expectations of what the fruit should look like. But this is just showing you this as a kind of example of how perception in general is influenced not just by low-level retinal and early visual processing mechanisms, but also by cognitive and in some cases even emotional mechanisms. And actually I go into that in some detail in my final year option. Now on this slide, because I've finished talking about colour perception, I'm going to move on to the other topics in the lecture. I'm just giving you a list of other sources of information about colour perception. Of course you should use the textbook, but I think with vision it really lends itself to videos, because often having things being animated and lots of different diagrams can really help understand things about visual perception. So a couple of videos at the top that are more fun and superficial, and then if you want to know more about the physics of light, or more about opponent processing, or the famous illusion you might remember from a few years ago about the dress that could appear gold or blue to different people. It's exactly the same picture, but some people saw a blue dress and some people saw a gold dress. Why was that? Okay, now to actually get these links to work, you'll probably need to download the PowerPoint file from Blackboard. Now, in the next two slides, I'm going to switch topics and tell you the little bit that you need to know about what's called the two visual systems theory. And what inspired this theory was an attempt to explain the observation that in the brain there are approximately 30 different small regions, each of which contains a separate retinotopic map, a separate representation of the image on the retina. Now, why would you need so many retinotopic maps? Now, it is the case that each retinotopic map tends to have neurons whose visual receptive fields are specialised for a different visual property. So, for example, in V5 they're specialised for motion, in V4 they're specialised for colour, areas V1 and V2 are more to do with contrast. There are some areas that specialise in the depth perception information that we get from having two eyes. However, there are only about 4 or 5 basic visual properties. So why do you need fully 30 visual areas in the brain? Well, that led to this idea that there might be two semi-independent visual systems in the brain, one of which is called the ventral visual stream that runs along the ventral part of the occipital lobe down into the temporal lobe at the bottom of the brain, and this stream is thought to be specialised for perceiving the identities of objects, the identities of faces, and properties of those objects. What is it? It's the what stream. And then thought that information from the occipital lobe flows up into what's called the dorsal visual stream and ends up in the parietal lobe for a different purpose. And that purpose is to know more like the spatial locations of objects in the world. Where are they? And also to control our actions towards those objects. So, for instance, visual processing that allows us to shape our hand appropriately to pick up an object in order to scale it to the size and shape of the object. So more metric properties of the world. So that's the theory, and it's been quite an influential theory over the years. It's mainly associated with the names David Milner and Mel Goodale. There are various versions of the theory. In some of them, the two streams are thought to be very separate, and in others, they're thought to be more interconnected. In the next slide, we'll take a brief look at some of the main evidence for this theory. So, the evidence for the two visual systems theory, or the most famous evidence anyway, is the following double dissociation. And both of the four parts of this dissociation are to do with what happens when people suffer focal brain damage due to a stroke or other injury to the brain. So the first dissociation is visual form agnosia, and there's one particularly famous patient with this called patient DF who suffered carbon monoxide poisoning, producing ventral stream damage. Now that's highlighted in the brain diagram on the left, showing the lateral occipital area, which is the area that in patient DF is most damaged. And in the left it shows a healthy volunteer MRI scan during visual processing, that area becoming active. On the right, the lesion in patient DF. And patient DF has quite a striking type of visual deficit. She can't really see things. She's sort of blind, and yet if you put an object in front of her, she can interact with it with her hands. If you hold something up in front of her, she can reach out and pick it up, and it's quite a subtle intact ability that she keeps. So, for example, if you change the orientation of the thing you're holding up in front of her, she'll be able to adjust the orientation of her hand or adjust the shape of her grip very appropriately, and yet she doesn't see the object, and so she therefore is not very good at identifying objects. She can't really tell you what things are other than by using kind of indirect cognitive strategies to work it out. So that's what happens when you get damage to the ventral visual stream. So the second part of the dissociation that makes the evidence stronger is the evidence about what happens when you get damage to the dorsal visual stream. And there's a YouTube video that is quite striking that you should watch that explains what this damage is. This is called optic ataxia. Now, in optic ataxia, where there's damage to the parietal lobe, you will find that participants don't have the problem DF has. They can name objects, they see, they've got conscious visual perception, but if you would ask them to do the thing that DF still can do, to reach out and pick something up, they really will struggle. Often the hand simply doesn't form an appropriate distance between the thumb and forefinger. They'll often knock the object over or just completely miss and reach to a different location, despite the fact they can happily identify the object, tell you what it is, because their ventral stream is not damaged. So that's the classic evidence for the two visual systems theory. There is a lot more, but that's the one you'll hear about most. Of course, there's also evidence against this theory. It was perhaps more popular 10 or 15 years ago than it is now, but it still can be a useful explanatory framework sometimes. The last topic I'm going to cover today is another developmental disorder of visual perception, which is amblyopia. But before I explain amblyopia, I'm going to explain a little bit about what happens in the normal case. So, for any animal with two eyes that have a separation between the two eyes, and in our case it's about 6 to 7 centimetre separation between the two eyes, you've got two images of the world. And when you look at an object that's far away, in order to get a sharply focused image of that object, you turn the two eyes so that the optic axes of the two eyes are parallel, as shown in the image on the left. And where it says accommodation, that indicates that the lens is lengthened or relaxed, so there's less focusing power from the lens. But when you look at objects that are closer than about 2 meters, and particularly when they get closer than about a meter, in order to get a sharp image of that object on the eye, you need to converge the two eyes, go a little bit cross-eyed, well, not cross-eyed, but you start moving towards that cross-eyed position, if you look, which is called convergence. You basically create a triangle, as shown by those lines. And also, the other part of focusing on a nearby object is to fatten the lens in order to get those near objects focused, which is achieved by the ciliary muscles. So that's all well and good, but the brain has to do some quite clever stuff when you have two eyes, because of the fact you've got two images of the world. So it has to do something called binocular fusion, because you see a single world, you don't see two worlds, you don't have double images or diplopia. So how does it do that, given that the two images are quite different from each other? Well, it actually learns to map the position of the left image to the right image, or the right image to the left image, as you're growing up. And that's the process of binocular fusion, but it's not as simple as that. It has to be a dynamic process, because depending on the convergence state of the eyes, the actual mapping that needs to be done is mathematically different. The images are not always different in the same way. How different the left eye image is from the right eye image depends how much your eyes are converged. So your brain learns that mapping as you're growing up, and then you won't have double vision unless you decide to press on the side of one eyeball. Go ahead and do that now, and you experience double vision, because now you've produced a physical mapping between the two eyes that the brain has not learned or tuned itself to when you were young. And equally, if you drink far too much alcohol and lose the coordination between the two eyes, then you end up with diplopia too. So what happens in amblyopia is that one eye doesn't move properly. It moves either not at all or less than the other one. And the brain, therefore, is not getting the signals from the eyes it needs to learn how to do the binocular fusion at all the different possible angles of convergence that happens in the normal developmental case. So the child will be experiencing annoying kind of double vision through this, and the brain responds to that over the medium term by actually suppressing the neural signals from the eye that's not moving, which is often called the lazy eye. So you can actually end up with functional blindness in the lazy eye, even though the eye itself is functional, that the cortical representation of that eye is suppressed to the point of blindness or near blindness in that eye. And of course, we also find that because eyes are important for social signals, if you've got an amblyopic lazy eye, you can give out strange social signals, and it can be socially disabling. Currently, the best treatment we know for amblyopia is to patch the strong eye during childhood, because if you patch that eye, you force the lazy eye to work and overcome its weakness. And if you can get it moving properly and then do binocular coordination eye exercises, then you can ameliorate or partially overcome amblyopia. I'll finish with a summary of today's lecture. Colour vision is possible because the three types of cone have peak sensitivities to different wavelengths of light. We can perceive more than three colours because we have an opponent process based on subtracting the output of the different cones from each other. And you can perceive the same set of colours under widely varying sources of illumination because the brain is able to estimate the wavelength composition of the illumination and subtract it from the retinal input, and this is called colour constancy. In the brain, cortical visual processing is divided into two interconnected streams, the dorsal where stream or action stream and the ventral what stream. In development, myopia is caused by lengthening of the eyeball and hyperopia by shortening of the eyeball, and amblyopia is a functional or cortical blindness in one eye, and it's caused by a problem of binocular coordination during childhood.